Okay, so hello. Today I will with Martin present results of our project exploration techniques in latent space for generative models applied to antimicrobial peptide design. First, let's talk what are antimicrobial peptides, in short AMPs. So they are short sequences of amino acid. We can see on the right side that we can represent them as like a string of characters. Second important thing is that they kill bacteria, and that's why they are attractive alternative to antibiotic treatment. And because the bacteria gain resistance to AMPs much more slowly than to antibiotics. And this is promising solution to the threat of antibiotic resistance, which will cause in future, there are some predictions, large amount of deaths. And this is our goal, to discover novel and diverse antimicrobial peptides. There is a research in this topic, and HydrAMP is a model that is trained exactly to generate AMPs. It is conditional variational autoencoder, which is optimized to create meaningful, latent, real valued vector space, which are just representations of peptides. And we want to think about this autoencoder as a creation of map. So there is well-known manifold hypothesis that we leverage in our project, that real data lives in lower dimensional manifolds embedded in high dimensional space. And autoencoders can be thought as like embedding to low dimensional flat map. And of course, due to this fact, we want to use Riemannian geometry tools in our project. Our approach divides into two stages. First stage is global exploration. So on the whole map of our map of peptides, we want to find some peptides that we call seeds. And then we zoom in and explore locally to exploit this specific seed to find even more and even better analog peptides. And now let's jump into the first stage, global exploration.
Right. So in terms of this global exploration, we want to talk to you about our first technique, which is B-seed exploration. So in hindsight, our backbone HydrAMP is pretty good to analog generations, which means it generates peptides which are similar to the ones that he has already seen, but we want some other aspects that kind of differ from that we know. So what we do, we are taking two known peptides, AMPs, and in short, we want to connect them with paths and search if alongside this path we got some like very promising or good with good qualities peptides, which we can see visualized on this presentation. So this seed with red circle is one that we would like to see, which is between two known AMPs. We do not want to bore you much with formulas, but just to short and visualize what we mean by that. So this short path between two known peptides we will call geodesics, which is just the short path, and this is formula that we use to find this length of this path. Naturally, this is integral, so we need to approximate it. So we actually using like sum with some points on this path, but this geodesics isn't enough to like find anything. Isn't good enough to find good enough peptides. We also need to add some additional term, which we can see with this third formula. This formula penalizes our points, our peptides on these paths if they do not have very positive antimicrobial activities. In short, we penalize if they are not like very good for our fighting against some potential pathogens. And this first aspect just to penalize us if our path is very long, and second, kinda forces this path to have some good, potentially positive candidates along the path. We also a little bit changes with coefficients because it gives us a very good analog to some aspect in physics, which is called least action in physics. This first path, it just tells us that physics or in nature, nature always finds the shortest path with least amount of action. And this first terms actualize gives us analog between constraint on space and time, and second to minimal potential energy. So this is just for that this is not like random loss function. It actually have some analogs in our physics theory. Okay, and to visualize, so we got two potential peptides, and this red line is just the simple shortest path along some globe. But we might actually want to like traverse via this purple line because we want to go from points that have like similar temperatures. We can think this as swimming across the globe, and we want to swim across oceans that have the same similar temperature. Okay, and these are some our results from this. So first chart is just simple path, and we can see that we are starting from some potential good peptides, which meaning they are their antimicrobial activity is like at the bottom of the chart, and but alongside this path we have like very poor candidates. But this optimized potential path gives us some potential candidates which we call promising seed in this chart. All right, and this is we're going to local explanation.
So this is second stage of our framework, which is about local exploration. And in this case, we use random walk to locally explore the proximity of a given seed. But of course, we are still in a Riemannian framework, so we don't want to treat a map as a flat surface. We want to be geometrically informed. So in simple case, random walk have two stages. First, sample direction uniformly. In Riemannian framework, it means that we sample uniformly according to Riemannian metric. And second stage is go straight for short time. And in Riemannian framework, it means go straight along geodesic. To compute these two stages in Riemannian framework, we have to compute local coordinate system because this is key idea in Riemannian geometry, that we can describe a geometry of manifold locally. And in this case, coordinate system can be computed as Jacobian of the decoder of our autoencoder model. And we can use, because anyway we have to compute it, and we can use those directions of our coordinate system as we can interpret them as mutations, because in ambient space, these directions means changing a peptide sequence. So we can think of them as mutations. And it means that from knowing coordinate system itself, we can already know how what neighbors are in the proximity. So we use random walks, and then in every step, we are computing coordinate system, and from the coordinate system, we can see what neighbors are in proximity.
All right, so Now we'll be going to our second approach, but we need some small introduction or reminder how Bayesian optimization algorithms work because we'll use it. So in very short, we want to have some other candidates from our known peptides and how we can do it. So we have some potential candidates. We need some oracle, which we call like black box function that is costly to use, but gives us some predictions how this potential candidate peptide will behave. But we do not want to use it very often, and actually we will penalize our approach if we use it. So we'll call it the budget, how many times we call this black box function. So we got some candidates. We are building surrogate model in our case Gaussian process. We searching some potential candidates with acquisition function, which is based on which tries to like simulate what this black box function would predict, but is like not so costly to use, so we can use this on those candidates. And based on those best candidates from this acquisition function, we choose next peptide to our pool that in the next step we'll use to build again next surrogate model. All right. So this is a table with results that we got. So this first method, so our Bayesian optimization, Riemannian random walk mutation, which Adam talked about earlier. And we got some other baselines from a repository that we tried to set up from GitHub. Those are methods that we like code with our own hands, but they were already made, so we only set up for our particular projects. They differ in this step explore. So in this explore step, we actually using this Riemannian wolf mutation. They are using other methods like trying various domain of those random mutation, genetic algorithms, diffusion sampling, etc., etc. And we compared this with this antimicrobial activity. The lower, the better in our case. So, and we are doing this against four starting peptides. This first peptide, FL14, is actually peptide which happened to be from this BC technique from which I was telling earlier. And we got two tables in terms of this evaluation budget because we can compare this Bayesian optimization only in terms of this evaluation budget. So calling this black box function oracle. So we can see that when the black box evaluation budget is relatively small, or like 150, in 2 of 4 peptides we get the best results. But if our evaluation budget was much more higher, well, 1,400, then we in 2 peptides that we actually checked, we got the best antimicrobial activity. Okay, so conclusion, we give you we wanted to present these two techniques. So first one was to like based on 2 promising candidates find the first some other candidates alongside the path, and we are actually really we think this gives us very positive results. And second was this integrations to Bayesian optimization, our techniques. So so in terms with comparison with other baseline, it also gives us some promising results, which we want to try and find explore more further. We are also very happy with the fact that our techniques are model agnostics or modular, so we can actually use it for other aspects, but we are focusing on this peptides exploration. And that's mostly it. Thank you very much.
Any questions? Well, I have a lot of talking today. So I'm curious whether you tested that, for example, for this exploration procedure, simply a random mutation, not a random walk, but random mutation, because if I understood that correctly, random mutation is like you simply apply a mutation, then you run the model and check whether it's better or not, right?
Yes.
But you can also randomly mutate peptide for a search.
Okay.
run your VO?
So if I understand well, we used this random mutation. Okay, you talked about random mutation, right?
Mhm.
Okay, so is this a good idea, like a baseline, right? To have our Bayesian optimization, but with another exploring technique, like of random mutation. This is very cool idea. We didn't check it. We are happy to check that. So it again differs in this explore step, right? We don't do it like in smart way. Use Riemannian framework, but you proposed just to randomly mutate. Okay, nice idea for a baseline.
Any other questions? If not, so thank you once again.
Thank you.